Hello, everyone. Welcome to this AnaBios webinar. My name is Chris Mathis. I'm the Chief Commercial Officer at AnaBios, and I will be moderating this session. Before we do that, I have a couple of announcements, and I'd like to go through a brief introduction of AnaBios's hepatocyte offerings. So my first announcement is that next in our series of webinars, we have 3 pain webinars in September, October, November. They're shown here. And the first is by Dr. Rajesh Khanna in September. The second is by Dr. Vladimir Yarovoy in October. And the third is Dr. Anna Moreno. These are all about pain. I know that may not be the topic of interest for this audience, but I'm sure you may know someone involved in pain research. It's a very important area. So please let them know, and they can go to our website to register. All right, so before I introduce Dr. Vernetti, I'd like to tell everyone about AnaBios's hepatocyte offering, and you can get a copy of this deck by sending me an email, which is shown here. And by the way, if you have any questions, you look at the panel on the right, and there's a questions box. You can write those in anytime during my presentation or Dr. Vernetti's presentation. And you should also see in the handout section that you can download our hepatocyte brochure and also the certificate of analysis for the lot that I'll be talking about in my presentation. So I think that many of you may not know AnaBios. We've been around for over 10 years. We're based in San Diego, which is shown here on this map. And over those 10 years, we've developed a very extensive network in the US only of hospitals, transplant centers, and organ procurement organizations, or OPOs, that allow us to receive human organs for research. And these are all consented for research. Now, obviously, the priority of organ donation is transplant. But if transplant cannot happen for logistical or medical reasons, then we are able to obtain organs for research purposes, even commercially research. So this shows the process. So we at AnaBios are able to obtain human brain, heart, lung, liver. This is a neuron representing dorsal root ganglia, which is important for pain research, and spinal cord. Obviously, liver is important for this presentation and is part of our hepatocyte isolation process. We have developed proprietary processes during transport that minimize ischemic injury. We've developed proprietary solutions and processes to provide very high quality cells. And what makes us different from a tissue bank, which we're not, we're a preclinical CRO, is that we're able to isolate cells and tissues that we use for functional assays. For example, we have cardiac assays, and we have calcium imaging electrophysiologies for pain research. So that the proof is that of quality is the fact that we can do physiology experiments. Another form of proof is our high RIN scores. So you'll see on our website that our RIN scores are typically 8 or higher, often 9 or 10. And whereas if you obtain human tissue samples from a tissue bank, they could be 7 or less. This is especially important for transcriptomic analysis, and again, points towards the high quality of our tissue samples. And by the way, in our archive, we have over 20,000 samples, human tissue samples. And at the moment, we have a promotion to enhance translational research. So we're offering some of the tissue types, heart, for example, liver, DRG, and spinal cord, at a highly affordable price. So if you're interested in obtaining tissue samples for your translational research, please contact me at the email I showed at the beginning, cmathis@anabios.com, or send an email to info@anabios.com. You can also write a request in the chat or in the question section. So obviously, AnaBios has one of the best lab teams in the business as a CRO, as a preclinical CRO. Another thing that makes us different from human tissue companies is our industry-leading clinical team, which has over 40 years of experience in developing protocols for recovering organs and tissue for research to optimize drug discovery. This dedicated clinical team has combined decades of clinical knowledge and clinical pathology experience. Especially for hepatocytes, they are trained to identify healthy livers for recovery. And this is important for the hepatocyte isolation method. So what I'd like to do for the rest of this presentation is to talk about our comprehensive hepatocyte certificates of analysis. I'll kind of give you an overview here on this slide, and then take a deeper dive into one of the lots that is spheroid qualified that was validated at one of our customer sites, and I'll show you some of that data today. So this shows an example of a certificate of analysis. Again, you can download one in our handout section. We show batch information, viability, cell count, confluency, number of vials, functional activity, metabolism, induction, transport activity, suspension replatable, 3D spheroid competent, for example, and extensive donor demographics such as age, BMI, sex, race, drug smoking use, for example, and cause of death. We also provide a detailed serology report. So the AnaBios advantage for quality hepatocytes is that it begins with qualified clinical assessment. It's enabled by proprietary recovery and isolation methods that we have been working on for the past 10 years and really have become the best in the business in this area. It's verified through functional and assay relevant QC, and we provide additional support to ensure success. So let's take a look at this particular lot that is spheroid qualified. Again, this is the COA that you can download. It has basic information such as the post-thaw viability, in this case 90%. It contains a minimum of 10 million cells, post-thaw confluency at 95% on day 5. It has donor demographic information. And very important to this process, those of you that know hepatocytes well, are pictures. This shows 5 days post-plating, 14 days post-plating. This shows cDFDA stain of canaliculi formation. And of course, the ever important spheroid formation after 11 days post-thaw. Then on the second and third pages are detailed information about enzyme, induction activity, metabolic activity, and transporter activity. And of course, on the last page is the detailed donor demographic information. Now I'm showing you this particular lot for 2 reasons. One is that spheroid qualified hepatocytes are really ideal for microphysiological systems that we'll hear more about in Larry's presentation. And also, as I mentioned, one of our customers has validated this. Actually, several customers have validated this lot. This particular customer has given us permission to show some of the data. So in this case, the customer has a benchmark hepatocyte. The ID is A, and they're comparing it to this AnaBios primary human hepatocyte, lot 1045. And these are the conditions and data assessment on the right. The first was 2D collagen-coated plating. The second was 3D aggregation. And here's a list of the data assessment in this detailed validation study. They looked at the total number of cells per vial, the number of recovered cells, cell viability and cell morphology, 2D plated viability cell morphology, 3D aggregated spheroid formation for 5 days, CYP activity, and other criteria that I won't be showing all of this today. This slide shows the summary of the evaluation. Again, this is the benchmark from the client that they compare to all of the hepatocytes that they look at, and compared to the AnaBios lot that happened to come from a pediatric donor. Expected viability in our lot was greater than 90%. The tested viability was 85%, which is quite close. Expected cells per vial was 10 million, and the tested cells recovered was over 11.5 million. So the recovered cell counts and viability were right on target. The AnaBios hepatocytes also passed viability tests. This slide shows day 5 viability. The cell viability of 3D spheroids was measured using calcium AM live and propydium iodide dead stains. So viability was high for both lots tested, and core death was not observed. This slide shows that the AnaBios hepatocytes passed plating criteria. This slide shows the 2D plating at day 5, and all cells were seeded at specified seeding density and maintained for 5 days on a 24-well collagen-coated plate. So the monolayer integrity, polarization, and biocanaliculi formation were both observed for both lots. And very importantly, the AnaBios hepatocytes formed spheroids as promised. This slide shows 3D aggregation at day 5. All cells were seeded at only 100 cells per microwell for 5 days on a 24-well agar-well plate. So lot B, the AnaBios hepatocytes, was not quite as spherical as lot A on day 3, but spheroids formed after 3 days in culture and became more rounded by day 5. You could even say that the AnaBios hepatocytes formed a spheroid that looks like a microliver. And for CYP activity 3A4 was measured on day 3 and day 5. This slide shows a comparison of the customer benchmark versus the AnaBios hepatocytes, luminescence versus days in culture, and whether 2D or 3D. And the CYP activity was comparable between the two lots and was higher in 2D cultures by day 5. And this may have occurred just because of donor-to-donor variability. Finally, I just want to make sure everyone's aware that earlier this year, Cell Systems became part of AnaBios. And Cell Systems has been around for over 30 years and is well known for providing high-quality primary cells for preclinical and academic research. And particularly important for this webinar is the primary human liver sinusoidal endothelial cells. So these are also very important in combination with hepatocytes for liver chip technology. In fact, Emulate uses the Cell Systems LSEX exclusively. You can see that in this paper. And yeah, they're quite important, and you can purchase them from the Cell Systems website, or you can send an email to customer service at cellsystems.com. And coming back to the AnaBios hepatocytes, just to wrap things up, you can go to the website and get more information about the AnaBios hepatocytes, media, and you can look at all the lots. In fact, this has lots of pricing information. This is the lot that we just looked at, and you can order them online for convenience. So again, if you have any questions about AnaBios hepatocytes, you can reach out to me by email, or you can put a question in the question box. So now it's my pleasure to introduce our speaker for today's webinar. Dr. Larry Vernetti is a research associate professor and group leader of drug safety evaluation in the Department of Computational and Systems Biology, Drug Discovery Institute at the University of Pittsburgh. Since 2012, he's been part of the team which has received NIH and EPA funding to develop microphysiological systems, or MPS. He's a member of several working groups aimed at a greater acceptance of MPS in the discovery and development pipeline. Before his time at University of Pittsburgh, he spent 15 years at Abbott Labs, and he did his PhD at the University of Arizona in toxicology. So it's my pleasure to introduce our speaker today, Dr. Larry Vernetti, the use of human liver cells in microphysiological systems. Larry, go ahead and take it away.
Thank you very much, Chris. Today I'm going to be addressing the use of there we go. Now we're active. Okay, today I'm going to be addressing the use of primary human liver cells in a liver microphysiology system and discuss topics related to their fit for purpose use as ADMET testing and liver disease modeling. I won't spend a great deal of time on this slide other than to point out that what anyone wants to build is a liver model with as many in vivo-like functions as possible. The typical published liver MPS model has anywhere from 2 to 5 liver cell types, which can be from primary cell sources, established cell lines, and of course, the use of liver-like cells derived from pluripotent stem cell sources. But just for clarification, I won't be discussing the use of embryonic or adult iPSC-derived liver cells today. That would actually be a topic on a presentation on its own. Key needs for MPS liver models are, of course, 3D organization, media flow, and cells that possess critical liver functions that respond to various chemical, biological, and molecular driver cues. Now, there are several types of 3D liver models, such as spheroids, which Chris has already talked about, and organoids along the same setup situation. But at the drug discovery laboratories at the University of Pittsburgh, we built a true liver biomimetic model. And my presentation will therefore focus on this liver biomimetic, and in a couple slides, I'll show you how to set up and kind of the design of it. We'll do a quick flyover with the liver cell types, although Chris has made an introduction to some of these that go into creating the MPS models. As a group, you probably all are aware of the fact that a primary hepatocyte is the parenchymal cell of the liver, and it's hard to tease down the number of functions that the liver performs with a hepatocyte, because there are over 500 vital functions that the liver does to maintain homeostasis in the body. It's hard to distill it down to a couple of bullet points, but what I have listed here are in the healthy liver, a few, and as well as some changes that are found when you get into liver diseases, such as the increase in steatotic vacuoles as shown in the bright field image of the liver culture. Over here, we have the stellate cells. That's a vital cell type, especially in the development of liver injury and disease, because it will transition from a quiescent vitamin A storage cell into a proliferative, fibrogenic, and contractile fibromyoblast. Now, the liver sinusoid endothelial cells, which AnaBios does have, they are a uniquely highly specialized vascular cell, very different from other endothelial cells that form the vasculature in the human body. The LSECs are highly fenestrated. Fenestrations are pores or holes which allow rapid passing of non-cellular blood components into the hepatocytes and the rapid uptake of hepatocyte products back into the bloodstream. And there is a loss of fenestration as shown here with the liver disease, and also it happens unfortunately with increasing age. Kupffer cells, of course, are the resident immune cell in the liver, and they are the first responders to intestinal endotoxins and pathogens as they fulfill their immune surveillance role. Kupffer cells are a tissue macrophage, and about 20% of the cells in the liver are Kupffer cells. And just as an aside, 80 to 90% of all macrophages found in the body are the Kupffer cells found in the liver. Now, the healthy Kupffer cells reside in an anti-inflammatory M2 or as a pro-inflammatory M1 state, but the ratio in the healthy liver of M2 to M1 favors the M2. However, that shift favoring M1 state is a key event in the initiation of injury or disease, and with a consequential increase in secretion of pro-inflammatory cytokines. The macrophages are, of course, bile duct epithelial cells. In the healthy liver, they transport bile acids into the bile duct and eventually empties into the intestine. Bile acids are necessary for digestion, and the bile duct epithelial cells also aid in the elimination of bilirubin breakdown from red blood cells into the intestine as well. Now, the impairment of bile flow by disease, drug inhibition, or by physical obstruction is a condition called cholestasis. And cholestatic disease treatments are one of the unmet clinical needs, but that will be beyond the scope of my discussion today. Now, the point in showing these cells in their healthy and disease states are to point out the various phenotypic and molecular changes which point to assays that can be used to monitor liver injury, liver disease, initiation, progression. There are many commercial and resource models available that have been published and are in use. All basically, of course, provide a surface to attach your cells and channels to direct media flow in and out of the devices. The differences typically are in the geometry, some specialized structure, and the materials that are used to construct the devices. Now, here at the Drug Discovery Institute, we built our liver biomimetics using either this, what I'm circling here, this microNet two-chamber glass device shown here, and next to it is a Nordis single-chamber device that we shown here. We try to avoid any devices having a high percentage of PDMS, which is a soft polymeric material, and unfortunately soaks up lipophilic compounds. So why do we build a biomimetic when there are other 3D models available, such as the steroids and organoids already mentioned? Well, first let me state, which is probably obvious to this group, is that at least in the in vitro setting, the liver is often just used as a drug metabolizing engine, but it's actually much more than that, because the liver, of course, plays a crucial primary role and important secondary roles in multiple diseases such as type 2 diabetes and metabolic syndrome. What we do at the Drug Discovery Institute is we're modeling our liver models based on the smallest functional unit of the liver, which is the liver acinar sinusoidal unit, as shown here in this cartoon. Hepatocytes run along the sinusoid, which is really an enlarged blood vessel, blood lumen, going from the periportal to the central vein region, as I just pointed out here. This is, of course, lined with the liver sinusoidal endothelial cells and the other key cell types as shown there. Now, as blood flows from the periportal region to the central vein, it creates what is called zonation, which in its simplest form is an oxygen gradient, which starts out here at the junction where oxygen-poor portal vein mixes with oxygen-rich arterial blood to form an area of high oxygen tension, which decreases as blood flows down the sinusoid towards the central vein. Typically, the liver is divided into 3 oxygen tensions or zones, as you see here, whereas the higher levels of oxygen are in zone 1 and the lower levels in zone 3. Now, there's more than just oxygen going on and a decrease of oxygen in the excretus, because these various zones in the hepatocytes take on differential functions. For example, albumin is produced by hepatocytes at higher levels in zone 1, whereas higher levels of oxidative drug metabolism are found in zone 3. Gluconeogenesis occurs at higher levels in zone 1 and glycolysis at higher levels in zone 3. Now, what's important here is in the area of drug-induced liver injury and the increase in steatosis, inflammation, fibrosis, and ballooning hepatocytes typically found with metabolic liver diseases, these usually initiate and predominate in zone 3 before spreading to other parts of the liver. So it was this need to recreate liver zonation that guided us in developing the liver biomimetic in lieu of other types of liver models. Now, over the past decade or so, we've evolved our liver models to take advantage of increased availability of primary non-parenchymal liver cells from commercial sources, the addition of new assay types, and new materials and new devices with alternate materials. So of the 3 models we've developed, I will be really focusing on this middle model, which we call our liver acinar microphysiology system, or LAMPS model. This is our second iteration, and the reason it was important to us was because in this model, we were able to actually recreate a zone 1 device by monitoring, by changing the media flow here, or create a zone 3 device so we could really specify the area of the liver that we wanted to look at and impacting drugs. And the third iteration was this, what we call our vascularized or VLAMPS model. This is a model that we buy from Micronet. It's a 3-ply model with a membrane in between, so it's a 2-chamber model. And what that allows us to do is that, in one chamber, we could flow whole cells through, such as peripheral immune cells or cancer cells, to evaluate their effect on our liver. And the second one was, this was a model that ended up being relatively easier and had some advantages in conducting the liver to a second organ model, such as I'll describe at the end of my session. Quick word on the various assays that we do on our liver MPS models. We divide the assays into 3 types. The in-life assays where data is collected during the incubation period by non-destructive methodology. We collect, in our case, we collect our in-life measurements using high content imaging and followed up with image processing analysis for quantitative data. We also collect secretome at various time points for various measurements, including the cytokine panel shown down here. And this is a way to monitor hepatocyte health, function, drug metabolism, and the release of injury and disease-specific metrics during the incubation period. And then finally, at the end of our incubation period, we will fix our devices and make various endpoint readouts, most often using a component-specific dye, such as this neutral lipid dye seen here, or by immunofluorescence using specific antibodies. Now, as part of our profile for toxicology, we do include a set of assays that are specifically designed to identify common modalities of toxicity. For example, we measure inhibitors of bioefflux, or that's inhibition of biosalt exporter protein. We also have measures of direct mitochondria inhibition, and we can measure the generation of reactive oxygen species or the release of cytokines as an indicator of inflammation. These are all included in our profile. I'll step back one step to describe what a biosensor is, because that is an important component, especially of our toxicity assay. A biosensor is a fluorescent protein that we insert the genomic or the gene sequence for that through retroviral transfection into our hepatocytes. When it gets expressed, it is trafficked to the correct area of the cell, such as the mitochondria in the case of our RLS biosensor. And in that microenvironment within the cell, if reactive oxygen species is generated, the fluorescent biosensor will go from a relatively non-fluorescent here that you see in control that I'm circling, and to a treated version right next to it where it grows bright green. Make a lot of advantage with the use of these biosensors. Now, these in vitro models have different capabilities, so they're best thought of as context of use, which is also sometimes called fit for purpose. These fit for purpose can be quite broad, such as what you would see with the liver on a chip, which you can use it for many, many things. But it's not favorable for other kinds of use, such as the high throughput format, where a simpler model might be the better one. So the rule of thumb when choosing model to use is go with the simplest and cheapest model that fits whatever needs that you may have. Let's move on now to a common question I get all the time, which is how good are these models for predictive toxicology? You know, basically you collect a lot of data, but how do you use it for predicting clinical liver toxicology? Well, very few of our complex liver models rely on a single measure meant to predict human clinical toxicity. So what I thought I would do is present several examples of the analytics employed to make clinical liver toxicity predictions from multiple endpoints that are measured. Now, in my lab, we take up to 20 measurements over the time course of a tox study, which is typically 10 days in length. And of course, these 20 measurements are made up of in-life, secretome, and endpoint readouts. We tried several methods, including this rather simple summation of significant events, all the way to more dimensionless reduction tools, such as the ToxPy, shown here to generate a single metric. I am aware of another liver biomimetic group, ADMET specifically. They use a method that I'm going to call the most sensitive assay method, which is an approach that's described down here by Lorna Ewart at ADMET in their last year's publication in Communications Medicine. And now what we are now working on at the Drug Discovery Institute is we're starting to combine our in vitro data with predictive computational analytics to get a handle on a better predictive tool for clinical hepatotoxicity. Now this is not going backwards, there we go. This is an example of an odor concordance method. It didn't imply any advanced computational analytics, just simple statistical analysis to determine changes that were significance. We no longer use this, but when we did, it was simply summing up of all the assays, the number of cumulative adverse responses in the LAMPs and related to the number of FAERS incidences per 10,000 prescriptions, as you see here. And with these compounds, which I've divided over here into known hepatotoxins or accepted hepatotoxins, daily compounds and non-hepatotoxins, when you do a plot, you get a pretty decent R-squared value for that model. The emulate model is their most sensitive assay method. And in this case, they do test 4 or 5 different assays, as you see here. And then what they do is they take the value with the lowest calculable IC50, they do correct it for protein binding, and then they normalize it to Cmax to determine the margin of safety of the compound. Then this ratio metric method is then used to make a decision by essentially drawing a line in the sand, and any margin of safety greater than 50 would be considered non-toxic. any MOS less than 50 is considered toxic. And they actually published a very nice 87% sensitivity and 100% specificity using this method. So that's pretty good. My final example of how we're now starting to use the data is the use of combination of our in vitro experimentation with much more sophisticated computer analytics to predict the clinical hepatotoxicity. This is an example of what we published last year in Internal Journal of Molecular Science. This is in conjunction with our collaborator, the DeliSim services at Simulations Plus. And the result I'm showing here is tocilizumab. And tocilizumab is an IL-6 receptor antagonist. It's a humanized monoclonal antibody, so it's a large biological drug, but it does come with a risk of hepatotoxicity. So step one was we ran tocilizumab through our LAMPS model, and of all the measurements that were significant, what we found was there was an increase in RLS shown here, and there was an increase in steatosis. And there was also an on-target effect of tocilizumab, which we measured, which was that IL-6 high levels of IL-6 will inhibit CYP3A4 activity, but tocilizumab effectively reverses that. So using the PBK simulation, which DeliSim did, they were able to take the known clinical Cmax plasma concentration of tocilizumab and simulate what the actual liver intestinal concentration was. And then they would use it in their biologics predictor, and they could then start predicting elevations in vivo of ALT. And what they found was that the steatosis and RLS measurement would indicate that ALT could go up in various patients, but that just the reversal of IL-6 CYP inhibition did not. Each of these symbols represents a simulated individual with the colors corresponding to the same individual. And what this data has demonstrated is a proof of concept that the data from a microphysiological system can be successfully integrated into a quantitative systems pharmacology model to start identifying liver liabilities, in this case of large biologic molecules. Okay, I included this slide for a couple reasons, but primarily to illustrate the performance of some of the methods that I just described. First of all, there are some pure in silico models using molecular descriptors. Some of these are older versions of these, but the bottom line is their sensitivity and specificity really not that good. But as you increase the complexity of the models to 2D down to the 3D models like the spheroids already mentioned, the emulate in the University of Pittsburgh biomimetics, you get a better predictivity, but the expense comes up the number of compounds you can effectively screen through there. You'll notice the in silico can do it in the hundreds of thousands, and the more complex models have a lot lower ability to do high content or high throughput type screening as that. Okay, let's move on now into ADME testing in the liver. PK, of course, or pharmacokinetics, describes the fate of a drug in the body. It's used to determine safe exposure levels to achieve efficacy. Now, there are lots and lots of ADME testing protocols published and predictors that are available commercially and as research tools in liver MPS models and in non-MPS models. So what I'm going to focus on is what we've done at the Drug Discovery Institute. Over here on the left is a cassette dosing of 5 compounds that went in as a single bolus mixture into a liver MPS model. And then we could, of course, over time, and you see week by week, we did over 3 weeks, that we would monitor the disappearance of a parent compound and the appearance of specific metabolites, including those that are driven by phase 1 and 1 conjugation reaction here. Over on the top here, we have 2 time response curves. The one here is reflecting the disappearance of diclofenac and appearance of its hydroxylated metabolite in a are the LAMPS MPS model, and when it reaches steady state. And over here is more of the golden standard 1 to 4 hour hepatocyte suspension media testing. So this kind of data can be done, can be collected, and it can be dropped into a predictive model, which I've used here is our PK predictor, which is in, oh boy, let's see, there we go. Is our PK predictor model, which is you can use through the biostatistics I'll mention here. And use of this PK predictor model will give you such PK parameters such as the intrinsic clearance, Cmax, T1/2, and things along those lines. And again, if you're interested in this kind of a PK predictor, I encourage you to look at the biostatistics column here. Okay, now we do measure bioefflux, and just because sometimes these movies that you see moving, they don't loop back to the start, I'll quickly say, please look at the movies and look at what's going on with the vehicle control versus the inhibition one of triglyceride dione. Oh, we lost our vehicle control. Well, if you look at it good, you would have saw that there was some green worm-like things appearing and disappearing over the course of 6 hours when the video was taken. And what that is a tracer dye that uptakes into the hepatocyte, metabolized to a fluorescent molecule, and then transported out of the hepatocytes into the bioconnicular spaces, which forms between the junctions of two hepatocytes. And that's bioefflux. That can be inhibited. You'll see in the triglyceride that is still running that we don't get much being pumped out of the cells. It gets sequestered in the cells. Of course, if this were a bile acid, that could be up to a toxic level within the hepatocyte. Over here, because these are collected by high content imaging, we can do analysis on them to make various quantitative calculations, as you see here in the terms of dye intensity and dye efflux. I'm going to switch now from ADMET talks and how we use our liver biomimetic model to study various liver diseases. Most of my time will be spent on MASH LD, and there'll be a short nod to type 2 diabetes at the end of my presentation. Okay, a clarification. What used to be called non-alcoholic fatty liver disease is currently being rebranded, mostly by clinicians, to a more precise descriptive event going on. And it's now being called metabolic dysfunction associated steatotic liver disease, or MASH LD. I apologize if I slipped. I'm used to the old nomenclature of NAFLD as compared to MASH LD, but it is a thing now. And later on, I'll also mention that what used to be called non-alcoholic steatohepatitis, or NASH, is now being called metabolic dysfunction steatohepatitis, or MASH. Okay, the models that I'm going to be describing is we took our MPS model and we could create a progressive disease model with it. Also briefly describe some pathway-associated predictive for drug treatment and get into some genetic polymorphisms, and then a quick word about type 2 diabetes at the end. Now, MASH LD is a progressive disease with an estimated 1/4 of the world's population suffering from it. In the United States, it's actually a little bit higher. It's probably a little bit greater than 30% of the population, mostly in older Americans. Now, although it is a progressive disease, only about 1/3 of those diagnosed with MASH LD will go on to develop more serious MASH, which involves not only steatotic hepatocytes, but also inflammation and early fibrosis. Now, a portion of these MASH people will go on to develop cirrhosis, and others go on to developing hepatocellular carcinoma. Both usually are end-stage diseases. At the Drug Discovery Institute, we have developed a model of MASH LD so we can test drugs and drug combinations and look at the reversal of MASH phenotype triad of hepatic steatosis, inflammation, and activation of fibrosis. Unfortunately, the prevalence of MASH LD is increasing in the US and worldwide. And in the US, at least, we have no FDA-approved treatment, but that's likely to change here soon. There are a couple of promising drugs that probably may be approved by the FDA within the next half year to year. But the lack of drug approval is not because pharma hasn't been spending time and mountains of cash to develop these drugs. The problem is that the history with developing this disease is that there were many in the hundreds of failed clinical trials. What typically happens in the clinical trials is that a few patients will have some relief with mast LD with the drug, but the number is small. For a recent example, obeticholic acid was recently not approved, although in clinical trials they did see some improvement with obeticholic acid for liver fibrosis over placebo, but it was limited to a very small effect size, as well as only found in about 1/4 of the subjects. And the cost of a drug like that was prohibitively high, so it was not approved. Okay, what we have developed in our liver model is that we can initiate and progress mast LD to an early MASH stage over an 8 to 10 day incubation period. We do that by two things. First of all, we do use a zone 3, our zone 3 conditions, oxygenation and gradient. And we use that because you remember that is the predominant site of the phenotypic changes found in many clinical liver diseases. And we force the disease down towards MASH and mast LD by use of 3 specially formulated medias, which mirror important components of serum at levels found in normal, early metabolic syndrome, and late metabolic syndrome patient blood. Up here is the timeline that we do this in this flow chart. I'll get to the actual media formulations on the next slide, but during the course of treatment, we do monitor everything that you see here in the way of disease metrics, both functional as well as phenotypical. And here is the media formulations that we use to maintain a healthy liver or progressed MASH LD. We start with our normal fasting media, and we then add to that increasing concentrations of glucose, fatty acids, and insulin, and we to form what we call our early metabolic syndrome media. And then if we increase the glucose concentration even higher, and then add in pro-inflammatory TGF-beta and the endotoxin LPS, we can drive the model to an early MASH with our late metabolic syndrome media here. Here is a summary of the kind of results we find using these 3 media formulations. The 3 key phenotypes again are steatosis, and in this case, with our early metabolic syndrome media and our late metabolic syndrome media, as shown in these pictures, we do see an increase in the amount of steatotic vacuoles within the cells. We can actually subdivide them down. That's I'm circling here in microvesicular steatotic and macrovesicular steatotic vacuoles. But the important thing here is that with our EMS and our LMS media, we do see an increase both in the intensity as well as the size and the number of them. We also then look at the stellate cell activation. Again, this we look at in 2 ways. The first way is through direct imaging using an alpha SMA antibody. It increases in both our early metabolic syndrome and late metabolic syndrome media, quantitated here as you see. We also monitor fibrosis as a secretome product, collagen 1A1 release, as well as TIMP-A1 release, and they both go up significantly treated with EMS and LMS disease. Then finally, we do monitor the cytokines that in our models, and those pro-inflammatory cytokines are increased in both EMS and LMS. This is work done by Mark Nito at the Drug Discovery Institute. Now, with this MASH model on hand, or MASH LD model on hand, we initiated a program to predict and test drugs and drug combinations. This is work presented by Dan Lefevre over here. What he did was he took the RNA-seq data from 182 patients that had been diagnosed with various liver states and generated a heat map that you see in the center there that showed gene expression and pathway clustering data for each of the individual patients. And that resulted in being able to classify, or cluster, I should say, the patients into 3 different states. There's a normal and steatotic state. There's a predominantly lobular inflammation state and a predominantly fibrosis state. This highlights is that distinct pathway expression profiles are associated with stages of clinical progression. Over here then, this is just a workflow, so it's not really hard data, but we use this workflow to associate the disease subtypes with their gene expression signatures to computationally predict and then prioritize, in our case, repurposed compounds, for testing in the liver MPS MAS-LD models. One of the drugs that was predicted from that workflow of Daniel's was of the HDAC inhibitor Vernetti, and it was predicted to reverse some of the disease phenotypes. So we then proceeded to test the combination of pioglitazone and Vernetti as a combination to reduce all 3 of these disease markers by monitoring over time what the drug treatment. And I'll get my arrow back. Okay, first of all, we looked at we looked at steatosis. Now, if you looked at pioglitazone on its own, which is not shown here, but we did look at it and published it in the publication at the bottom right of the slide, we did show that pioglitazone on its own will reduce steatosis, but it has no effect on inflammation or fibrosis. And Vernetti, which was also tested on its own, did reduce inflammation and fibrosis. It had no effect on, steatosis. But when we combined both of them together, we did see a reduction in all 3 phenotypes, such as we saw a reduction here in the lipid tox, which shows steatosis indicator. We saw a reduction in the expression of activated stellate cells through SMA staining. We also confirmed that with looking at secretome products of early fibrosis response with collagen 1A1 and TIMP1 readouts. And then finally, we did see a decrease in the secretion of the pro-inflammatory cytokines in this deal. So this was a real nice proof of concept exercise using this drug combination to halt the progression of MAS-LD disease. I'm going to finish up here the MAS-LD story with a little description on the known gene variants in the patient population. There are the variants, the PNPLA3, the most common one cited, that puts people with that variant at a higher risk for MAS-LD. And by the way, PNPLA3 gene is involved in triglyceride regulation. So there's a polymorphic mutation, the PNPLA3GG mutation, and we're going to compare that against PNPLA3CC, which is the wild type gene. Now, this is work using primary genotyped hepatocytes that were tested that had either the PNPLA3GG high-risk variant or the PNPLA3CC wild type variant in our LAMPS model. What Mabube and Ming Ying, who also goes by Summer, found is that the GG variant had significant higher levels of, shown here, lipid accumulation, shown here in by high content in the imaging, and then the analysis down here showing that the GG was in all 3 of our immediate types at a substantially increase in steatosis. The same picture is also shown in the fibrotic stellate cell activation, which is shown over here through high content imaging and quantitations for the alpha SMA expression. The GG again showing higher levels in all 3 medias. And then of course, the over here is our secretome measurements showing that we did indeed show that there was an increase in the steatosis with the GG variant. So this was a really good way of starting to do hypothesis testing as to why are these patients at higher risk. And we first showed that they are at higher risk, and then you could start to tease out more questions. And the final collaboration and data slide I'm going to discuss is an ongoing collaboration that the Drug Discovery Institute has with the Chemical and Petroleum Engineering Department at University of Pittsburgh, where we have a collaboration to link our MPS liver model to their model of the primary pancreatic islet cells to study type 2 diabetes. What they're showing here is that under disease progression, in this case just using EMS, not the LMS, they do show in the liver, expected that we have an increase in lipid tox or steatosis, an increase in the activation of early fibrosis through alpha SMA staining. And they also measured IRS2, which is an indicator of insulin resistance, and showed that IRS2 goes down, which means that insulin resistance went up. They also measured some important hepatokines, found that the liver disease media do increase various hepatokines like HGF and RB4. In this model, we didn't use the lamps I've been talking about. We used our VLAMPS model, which is the 2-compartment model, and they were then able to link the 1-compartment outflow from the liver directly into the pancreas and make various measurements as shown over here in the pancreas being the pancreatic islet culture. And in this case, it produced a loss of insulin response in the pancreas with changes in these secreted amylin, GLP-1, and pancreatic peptide, the PP. And over here are some pictures showing that there's a effect on insulin responsiveness. This pictures are on the top is a non-fasting. Is a pancreatic islet in a non-fasting media, and underneath it is the EMS media. Okay, my final slide is to go back. I mentioned the Biostatistics database a couple of times. I wanted to reintroduce it because it really is a really nice tool for data management, to optimizing experimental design, result analysis, calculating experimental inter and intra-lab variation, reproducibility. Computational tools for like predictive PK as showed, and some therapeutic modeling, has additional capabilities as well. But I would encourage anyone that would like to have a database capable of those kind of things to look at this Biostatistics website down here. You'll be able to see access to some of the kinds of data that I've shown today, as well as with other organs. Yeah, I'm going to summarize this now in 2 slides. First one, just kind of review what I showed, that structured MPSs are important in the continuum for fit for purpose, and primary hepatocytes are still preferred for ADMET studies. That when we can use this model for the MASLD, we do recapitulate those important phenotypes of steatosis, inflammation, fibrosis with our 3 media formulations. Drugs predicted from patient gene expression signatures were shown through a very nice proof of concept to actually decrease the disease metrics in the MPS models, and that you can use genetic polymorphic primary hepatocytes from donor livers to show, in our case, we show the increased risk of MASLD. My last slide really is what I call best practices for ADMET tox testing, and I suppose you could put in liver disease modeling as well. First of all, I'm a big fan of primary cells, hepatocytes, stellate, and L-sex, because they have fully differentiated functions, and they can faithfully recapitulate cell-to-cell tissue interactions. The use of scaffolding proteins is absolutely essential, both to support the function model as well as the 3D structure. Controllable fluid dynamics allows you control over oxygenation as well as replenishing nutrients, removing waste, and adding drugs in for that matter. I would encourage non-PMS device housings to prevent loss of lipophilic drug loss, and that we're starting to find out that the combination of in vitro experimentation with more sophisticated computational tools help not only remove bias and improve inter and intra-laboratory reproducibility, but build a better predictive outcome model. And with that, I leave this slide up. It's, I think, time for questions.
Larry, thank you so much for an excellent presentation and overview of the MPS field related to liver biology and liver studies. So we do have time for questions. You can write your question in the question box in the panel. And I'll get started. Larry, did I understand correctly that the devices you mentioned, were those developed at the Drug Discovery Institute, or are those commercially available?
Those, thank you. I did not clarify that. Those are commercially available.
Okay.
We use of commercial ones. We tried several of them, and these were the two that we settled on for our primary workhorses.
Okay, excellent. Here's a technical question. This relates to the efflux slide that you showed about halfway through with the triglyceride block. Is the fluorescent dye specific to MRP2 or BSEP?
Really good question. That's somebody that knows. It is specific to MRP2. It's not a specific BSEP inhibitor. Fortunately, that two, by inhibiting one, you typically inhibit the other one.
Okay, thank you. Here's a question from the audience. In the studies in which you model the transition into early and late MAS LD, how many different donor batches did you use?
Well, we've used several now, but the original publication was made on one donor batch. We have followed now, and oh wow, with everything, 5 or 6 at least, with and maybe even more. But 5 or 6 have been looked at in great detail, and a couple more have just.
Interesting. And did you observe any significant differences between those donors?
Yes, there are. There are some donors that achieve higher levels of mitosis and higher levels of fibrosis, and those that show lower levels typically are all significant differences from our normal fasting media.
Okay, and from the same person, what was the time of exposure to the different media that was required to produce those observed effects?
Okay, after we build our healthy liver model, from day one, we start flowing through our disease-specific medias, and then we do collect the secretome over time and make any in vitro in-life measurements that we need to make. But it's really by day 10 that we put the data together and show the increase in fibrosis and steatosis and secretion of the pro-inflammatory cytokines.
Okay, thank you. Now I know that you're involved in several working groups aimed at a greater acceptance of MPS in the discovery and development pipeline. What do you see as the major or biggest challenges in more widespread adoption of MPS in drug discovery?
That's a really good question. Yes, 2 things. There was some concern early on with the experimental variation or the reproducibility, not only within one lab, but the ability to reproduce a model in a different lab. And that's not a new challenge, but it's certainly one that needed to be addressed with widespread adoption. That's the reason why the biostatistics database is so good, because it can make those kind of calculations fairly easily, and made great advantage with that. Our model has been reproduced very well at Texas A&M, so there is some good intralab reproducibility with that. Other challenges, well, of course, these models, as good as they are, they don't recapitulate whole body physiology, but that's something people are working on.
Okay. Here's another question from the audience. Have you looked at the expression levels of enzymes and transporters in your NAFLD model? And do they correlate with the actual clinical data?
The answer to that is we have looked at some expression levels. My problem there, and I don't want to go off on a soapbox, but mRNA expression levels, especially with cytochrome P450 enzymes, is not really a good estimation of their activity. I'll stop there and say that yes, we have looked at them, and do they correspond to clinical levels? I don't know, but I will say that the activity assays that we've done, induction levels and inhibition levels, do have a pretty good concordance with in vivo activity.
Okay, thanks. We'll do a couple more and we'll wrap up. So what do you think about using donor mix versus donor match cells in MPS? And is matching necessary, and in what cases, if it is?
Great questions you're getting there. it would be preferable if you could get all 4 or 5 cells, whatever your liver model may have, of autologous donor cells.
Mhm.
That actually have made attempts to do that, but it's really
challenging. Yeah.
challenging.
Yeah.
Yes, AnaBios would be well aware of this.
Yeah.
If you can find donor match, there's probably very few vials of one of the cell types compared to others. So it's an ongoing issue. Probably one of the reasons why people like the concept of using iPSC cells. You can get autologous cells that way. Of course, I have problems with iPSC cells because they're not fully mature, but that's a different conversation.
Okay. And last one is, how is your MPS system standardized to maintain consistency from batch to batch during preparation?
Good question again. Mostly we depend on historic levels of our albumin synthesis, BUN synthesis. Of course, we have to follow a very strict protocol so that our models are made identically each and every time. And we get pretty good reproducibility, even months and a year apart, using this, as long as we use the same lot of cells. Every time we change the cells, we do have to kind of go through that whole process again.
Interesting. Yeah. Very good. Well, Larry, thanks again for an excellent overview and presentation. And for those of you in the audience that joined late or missed some, or you want to go through some detail, this will be made available for you online. And again, you can download the brochure or the C of A in the handout section. And thanks again. Stay tuned for more hepatocyte liver-based webinars from AnaBios for the future. Thanks again, Larry. Appreciate your time.
Bye-bye.
Thanks, everyone.